Okay, well, I'm going to switch gears a little bit and try and give you one little vignette to try and explain from my perspective as a mouse immunogenomicist come lately to the human, why I think the human cell atlas is going to be important for autoimmune diseases. So just to remind you, something that's probably not too appreciated, but over 10% of people have an autoimmune disease, but they're broken up into as many as 100 different diseases, some of them very rare, some common. And according to Tony Fauci on the NIAID website, the cost is actually twice that of cancer because most of these diseases are chronic. The problem that we have is that we really don't have a method at the moment for seeing the cells that are actually driving a given autoimmune disease. And I think that's where, and I'll give you one example, where some of the single cell technologies I think is starting to open it up. So at the moment, most diagnosis is done by specific antibodies in blood, a little bit of flow cytometry and some immunohistology or biopsies. But none of this really gets you at the cells to tell you what's actually driving the disease. Now, what we know about tolerance as a mechanism is that it's a whole series of checkpoints indicated on this slide by a series of red triangles that either suppress or kill off self-reactive cells either when they arise in the thymus or in the bone marrow or after they leave and go to the lymph nodes and spleen and blood. Now, many of these cellular checkpoints we've only really seen in transgenic mice. We still don't understand most of the genetic wiring that underpins them. And what's pretty clear, though, is that you can inherit in a mouse or a human a pretty severe lesion in most of these checkpoints and get along for years before you develop an autoimmune disease. Now, one of the ideas that we've been pursuing then is really that autoimmune diseases has common roots with lymphoid cancer and cancer more generally, that you have a series of checkpoints, and in fact what we know about the tolerance checkpoints is that both in cellular terms and in terms of the genes that underpin them, they actually do overlap extensively with tumour suppressor genes and tumour suppressive mechanisms. And so the notion is that that we have a huge burden of self-reactive lymphocytes, T cells or B cells, that are being generated both in the thymus and bone marrow or by somatic mutation in germinal centres, and then a series of checkpoints that are progressively weeding out the cells, each one of which may be a certain amount leaky. And so as a result, when everything's working perfectly, everything works fine, but if you have an inherited defect in one of the checkpoints, you start to have a large number of cells coming through and the other checkpoints then start to leak. And the notion is that eventually some rogue cells start to break through the other checkpoints because they acquire genetic mutations, perhaps the same mutations that arise that initiate the process of lymphoid cancer. But they could also be epigenetic changes. They could be changes in the way that the cell expresses genes. And the idea that one rogue clone might then feed off another rogue clone and eventually set up a stable and growing positive feedback loop, and that's when autoimmune disease. But we had no real way of testing it. It's all very nice to make up this story. So what I'd like to tell you then is one little vignette where I think we're just starting to, with very primitive versions of single-cell genomics compared to what many of you do here, are starting to crack that for one autoantibody. And so this is one of the oldest known autoantibodies, rheumatoid factor, originally discovered by Henry Kunkel back in 1957. And it's an IgM antibody that binds to IgG. So strongly enough that they form stable immune complexes in the circulation, and that's how Henry Kunkel found them. And later on with John Capra, he showed that actually these the monoclonal versions of these rheumatoid factors that arise in a variety of diseases seem to have recurring idiotypes, which just means now in genetic terms that they prefer to use particular variable regions on their heavy and light chains over and over again in different diseases and in different individuals. And we have no explanation as to why that is. IgG presents many epitopes, but for some reason it's all concentrated on one epitope and a few variable region combinations. And that's a mystery, and we don't understand what drives the evolution of these antibodies. So the disease that we've looked at to start testing this is Sjogrens syndrome. And one of the reasons for that is that it has this very interesting three phases. The first phase, which is an organ-specific autoimmune disease that affects the salivary glands and lacrimal glands, and it's troublesome in terms of dry eyes, but it's not life-threatening. But in a good proportion of people, they progress to a systemic disease where, in particular, these rheumatoid factors become pathogenic and form immune complexes in a variety of tissue beds, and that becomes quite severe. And along with that, there's a transition from a very polyclonal response, as far as we know, to a more oligoclonal response. So in this context, you start to see these uncle rheumatoid factors come up. And in a smaller subset of individuals, they progress to overt B-cell lymphoma. And interestingly, those lymphomas use the same VH-VL combinations and rheumatoid factors. And so that was one of the reasons we thought we might be able to test this, and in particular to see if we could understand, using single-cell approaches, what's actually happening in the evolution of an autoimmune disease and the origin of this systemic antibody. So to do that, we've had to try and pull together the old technology for how we diagnose disease, which starts with serum antibodies. And so with our colleagues in Adelaide, they've become very good at purifying autoantibodies to high purity and then sequencing them by peptide digestion and time-of-flight mass spec. The problem with that, of course, is that the really unique part of the antibody is the CDR3, which is unique in every different antibody, and you simply can't interrogate that by the current methods of mass spec. And so from the same blood samples, we take whole blood or peripheral blood mononuclear cells without any guess as to which cell type, if any, the circulating cells that are making this autoantibody might be in, make total RNA and then amplify heavy and light chain mRNAs and do deep sequencing. And that gives us a big lookup list when it's translated into a FASTA file that we can then interrogate the mass spec peptides and match up and actually generate then the nucleotide sequence that corresponds to the actual pathogenic autoantibody. In this case, one of these rheumatoid factors that are in a Sjogrens patient. And that then gives us the tools to go into the single cell analysis and say, can we actually identify in the circulation the single cells that are actually making this autoantibody and see what's gone wrong with them. So we're still really, this is a work in progress, but we've analysed about 6 patients at varying levels, and patient number 1 we're the furthest along, and I'll just show you a little bit because there's a couple of lessons in that. Now this patient has a classic kunkel rheumatoid factor, uses VH169 and VKappa320, and one of the nice things is that there's a very nice monoclonal antibody to VH169 that we can use for flow cytometric staining. So the plots that you can see there are 3 different time points in the progression of this person's disease, from an organ-specific disease before she had vasculitis through to very severe vasculitis in 2016. And what you can see on the x-axis, that's the staining for cells that are using this particular V region. Normally it's only about 5% of the cells. And on the y-axis, the kappa light chain versus lambda light chain, which is the other. Now we've already made a very educated guess as to where to look, and this is an interesting thing. In this case, that's worked well. We've gated on cells that using the classical markers that have been developed by people around the world for what defines a memory B cell, which are shown up there. And in this case, the rogue clones are in there. But in other patients where we know from deep sequencing of everything that the cells are there that are making the RNA that's causing the autoantibody, but when we've gated on this subset and done single cell analysis at the primitive level that we're doing, where we can only look at a couple of hundred cells, the cells that are making the autoantibody haven't actually fallen in there. So it's an interesting lesson of exactly what Christoph was saying, that you need the skydive. Because the minute we start thinking we're clever and know where to dig, sometimes you're right, and a lot of the times you dug in the wrong spot. So then, once we do know where to dig, we then, in this patient, we then sort the rogue cells, and from the same patient, cells that we know will be nicely separated on the flow from the rogue cells, because they use not kappa but lambda light chain, and don't use this V region. And then we sort them, in this case, just into 96-well plates, and also pools of 50, and the purpose of the 50 is to deal with the dropout problem that exists particularly on the DNA side. And then what we've done is G and T-seq, so that we can get from the same cell both DNA and RNA. The RNA gets processed through SmartSeq2. which allows us coverage of the variable regions of immunoglobulin as well as getting abundance of transcripts overall. And then with our collaborators at UNSW, Fabio Luciani's group has developed a very nice algorithm that reassembles the short reads so that we can actually get the complete heavy and light chain variable region sequence and any somatic mutations. From the DNA, we do multiple displacement amplification. In our hands, we only amplify probably about 30% of the template that's there, so there's a lot of allelic dropout, which is important at the single cell level, but there are ways of dealing with that. And then it's a standard Illumina library prep up 2 rounds of targeted capture for all the exons of 150 genes that are known from all the human lymphoma and leukemia sequencing to be recurrently mutated. So it's not a complete genome-wide sequence, but it's a targeted sequence. So this is what we get at the level of gene expression. Control memory cells and rogue memory cells from the same patient, analysed at the same time in parallel. So you can see the sorting's gone well. Most of the rogue cells, except for 2, are indeed using the V regions that correspond to the pathogenic antibody and kappa light chain, and most of the normal memory cells don't use that variable region, with 1 or 2 exceptions. And you can see then that there are many genes that actually differentiate between the normal memory cells and these abnormal memory cells. And they immediately tell us a lot about what's going on in these cells. What's really interesting is that many of them correspond actually to an emerging population of cells that's popping up in all different areas, both in mouse and human, of what are called age-associated B cells, or CD21 low B cells. They're marked with low levels of CD21, high levels of CD11C. So this is an interesting population that the field still hasn't come to grips with, and I suspect is, again, multiple populations based on the variations we're already seeing from one patient to another. One of the genes that's upregulated immediately has some therapeutic indications, and that's CD86. So it's much more highly expressed on the rogue cells than the normal ones. That's B7-2. It's the ligand for CD28, a very important co-stimulatory molecule for T cells. And many years ago, we did an experiment with Jim Allison that showed that simply dysregulating the expression of that marker was enough to completely allow self-reactive cells to bypass one of the main checkpoints that normally stops a self-reactive cell from getting help from T cells. So that immediately indicates one possible driver to this rogue state. And there are very nice blocking reagents that are in the clinic already approved for rheumatoid arthritis, abatacept in particular, CTLA-4 Ig fusion protein. So we're working with Bristol Myers Squibb to see how we could combine this kind of blood cell profiling where we can track the rogue cells with a clinical trial of abatacept and Sjogrens syndrome. Now, from the from the capture sequencing, what we found was that there were no mutations in any of the 150 genes except this one, KLHL6, and the mutation that was present in the rogue clone and not in the normal memory cells, so definitely a somatic mutation, is identical to one that's found in diffuse large B-cell lymphoma. It's a site of recurrent mutations right at the dimerization interface of this of the protein that it encodes, where it assembles this large ubiquitin ligase. So by going to all the single cell analysis, and this is I think my last slide, what we've been able to do is actually to see what the evolutionary tree is that's driving the production of this Henry Kunkel's rheumatoid factor in this patient. And basically there's a couple of messages. So we've been able to go back now and express the unmutated ancestor antibody that we don't find in the blood of the patient to see, well, what did it start out like before all the somatic mutations? And when we express that, it has undetectable binding to the autoantigen by ELISA. Now, it's quite likely that it has very low affinity, and once we go to plasmon resonance, we'll see very weak affinity, but we haven't done that yet. But then there's one big cluster of this evolving clade, you can see here, which is differentiated because of 2 key events that are highlighted there. A mutation in the heavy chain CDR3 that we think increases affinity, and this acquisition of this putative lymphoma driver gene, KLHL6 mutation. And that branch, when we express a representative member of that of the tree there, has intermediate binding to the autoantigen. So these mutations are doing it. But the other thing you can see in that tree is that you can see that the cells are rapidly mutating away from one another with, between 2, 5, even 10 mutations apart. So this is a very highly evolving, rapidly mutating branch of this rogue clone. And one branch of that has acquired a light chain mutation that we've shown experimentally further increases the binding to the autoantigen to be a high binder that's illustrated there. Now, so essentially acquisition of a lymphoma driver mutation seemed to be at the corner of a hypermutating branch of this clone. But then there's this other branch that doesn't have the KLHL6 mutation and has acquired the same light chain mutation independently and it already has high binding to the self-antigen, but seems to be very stable with very little ongoing mutation. So what exactly this means, whether there's another mutation in this branch or a mutation in a gene that we haven't interrogated earlier in the tree, it's early days. But the point of showing you, I think, was really just to say that even with very primitive technology like this, compared to what we're really going to, seeing rolling out now, I think we have a way to start to crack all autoimmune diseases and start to connect them back together rather than have them all fragmented as rare diseases as they are now. The first and foremost thing is that we have the opportunity, and this is where the atlas comes in, to be able to start to differentiate where normal variation in the aging of our various immune subsets departs into something that's diagnostically abnormal. And I think that's where we really need an atlas. We don't really know enough about what's a normal or an abnormal cell. Most importantly, and so we're really rolling that out, we're initiating as a program for 36 autoimmune diseases where we're collecting blood samples at the time of diagnosis before people have been complicated with a lot of heavy immunosuppression. One of the powers is that it really gives you this, if you can combine the gene expression with the immunoglobulin gene rearrangement, you now have clonal barcodes that allow you to look at the response to therapy, much the same way that we use those sorts of barcodes in minimal residual disease and leukemia or lymphoma. And more importantly, as I showed you there, we can start to see some of the common roots with lymphoid cancer and some targets for potentially more selective therapy to actually root out the drivers of the disease. The people that did this work, Joanne Reed is an expert in Sjogrens syndrome, senior fellow in my group, has worked closely with Tom Gordon, who does all the proteomics and clinical immunology in Adelaide. Manu Singh, a fellow in the group, has set up all the GNT-seq and all of that work. Catherine Jackson is a senior fellow, just come back from Stanford, an expert on all the bioinformatics of immunoglobulins. Our collaborators at UNSW, Fabio Luciani and his group, are really important for all the SmartSeq work that we've done. And I'll stop there, thanks.